We're about at 4:10, so I think we'll get going with introductions. Welcome to the second Spring Soil Science Seminar for Spring 2019. We're privileged today to have Dr. Marshall McDaniel from the department here coming to talk to us. And Marshall is an assistant professor in soil plant interactions within the department. His research focuses on the exchange of carbon and nutrients between plants and soils, and how management and the environment affect this exchange. Marshall teaches a junior and senior level soil fertility class, as well as a graduate class in plant-soil interactions and an agricultural travel class that travels to Argentina, Spain, and Australia. He also conducts outreach, parts of which engages farmers in collecting their own soil health data, and involves in citizen science and in getting farmers and producers involved in the process. And he strives to synergize research, teaching, and outreach to enhance agricultural productivity and reducing environmental impacts. So let's give a warm welcome to Dr. McDaniel, please.
Thanks, John. Well, thanks for everybody who came. I heard the plant breeding seminars at the same time, and unfortunately, I haven't even been to this seminar yet this year because that class that John was just talking about, the travel course I teach is right after this, and I'm usually preparing course material for that, but Hannah asked me to come give a talk, and I said I'll do it, even though I have to run to a class right after this, a couple buildings down. But anyways, thank you for being here. A lot of familiar faces, so we could do this informally. You can just raise your hand if you have a question at any time in this. But what I'll be talking about is participatory science, sometimes also called citizen science, and particularly doing it in soil science. So participatory science has gotten kind of famous through the use of smartphones. People are using it in ornithology or bird watching. They're using it in tree phenology, so timing of bud bursts and leaves forming on trees. But it's just starting to be used in soil science. And I'll be talking about some of the research we're doing in that area, and I'll put in some plugs for some other researchers I'm working with here at Iowa State. And then Teresa will be giving a talk, I think, later in the seminar series. So if you're interested in any of this stuff, come back and see Teresa's talk. She's going to be presenting some of her own data. I don't want to steal her thunder too much, so I'm not really showing much of her data. So I'm going to leave it up to you to come to her talk. And what time is your talk? Or what day?
March 6th.
March 6th, okay. All right, so I'm going to talk about soil health definitions. So soil health can be contentious. Everyone kind of has their own definition of it. I'm going to give you the one that I prefer. Madi and I might both have different preferential definitions. I use the NRCS. And then I'm going to talk about the reason for emphasis on soil biology and kind of make a case for that. And then I'm going to talk about soil health tests and why farmers might be interested in getting their soils tested for soil health. Talk about the ingredients of a good soil health test. And then I'll talk about the promise of participatory science, aka citizen science, in measuring soil health. And that's where I really want to focus on that in the theme of the talk, and then some concluding remarks and take any other follow-up questions. Okay, so like I said, everyone's got their own definition of soil health. It's trending now, and so are these commercial labs that are measuring soil health. But what is actually soil health? I like to use the comparison with human health, right? It might be difficult to define. In fact, I couldn't give you a very good definition off the top of my head, but we know what good human health looks like, right? There's indicators that we can go to. For example, body mass index, even though there's some controversy of that, we can get a human's body mass index, so we get their weight and their height. That's an indicator of human health. Temperature can be an indicator of health, a chronic sickness. If your immune system's reacting to a infection you have, you're going to be running a high temperature. Blood pressure gives us an idea of your circulatory system and strength of your heart. Pulse, kind of the same thing. So these are all good indicators of human health. But again, it's kind of challenging to come up with a good definition of what exactly is human health. So it should be no surprise that it's hard to define soil health too. But here's what I got, and I always encourage my students not to go to Wikipedia, but I happen to like this definition quite a bit. So I'm just going to read it to you. It's human health is the level of functional and metabolic efficiency of a living organism. In humans, it is the ability of individuals or communities to adapt and self-manage when facing physical, mental, psychological, and social changes in the environment. Is that what you would have said if I asked you what is human health? That's quite a comprehensive definition. So to compare to soil health, I put on the right the NRCS, which I prefer their definition. A lot of other folks do. So soil health, also to refer to as soil quality, it used to be called soil quality. I'll show some history and why that might be, is defined as the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, and humans. And then I've just highlighted some of these words and terms that are similar among both definitions of health, right? Function is in there. Sustain is kind of equivalent to adapt and self-manage. Continued. Living, of course, is in both of them. But soil health isn't really new, and I'll show some evidence from this from Google Ngram. But it's really a derivative and redux of the term soil quality before it, and I would even argue soil tilth before that. So when my grandfather was farming, they were talking about soil tilth. And really, health is separated with that term in living. So we're talking about only living things can be healthy, right? So that's the key part and key definition of soil health, right? And let me show you guys a bit of evidence. So how many of you have been to Google Ngram before? Raise your hand if you've ever heard of it. A few people have. So what Google has done, they've scanned all human literature going back to 1800. So not only are they reading our emails, but they're also doing some good things. They're scanning human literature. So this is a time scale. I know it might be tough to see all the way back there. This is just their, I took a screenshot of their homepage. It's from 1800 to 2000. And this, in the y-axis, is just the percent out of all the literature they've scanned that these terms have shown up. Frankenstein, Albert Einstein, and Sherlock Holmes. And it shows the trend over time. So these are very small percentages. So this bottom unit right here is 0.00002%. So it's a small percentage, but by looking at the relative change over time, you can look at trends and terms. So I'll give you the example of Albert Einstein that's shown on their front page. So Albert Einstein was born in 1879. So you can see his line, which is this blue one, hasn't even gone up yet. Of course not. No one's talking or writing about him. Then he published his theory of relativity in about 1908 or 1909. And then with a gap, kind of a lag time to get published and get established, then all of a sudden his name takes off, right? So that's what we expect with the data I'm about to show you, which is, of course, this talk isn't about Albert Einstein, it's about soil health. So what I did is I put the term tilth in, and now I've used my own graphs. I used their data and put it in Sigma plot here. And this is from 1880 to 2020, the scale here. And again, this is the percent. This isn't a very small number, even smaller than the Einstein use, I think, for the most part. And you see this trend of soil tilth kind of peaks in the late 40s and then starts to go down in use. If we look at soil quality, it starts out kind of slow and then starts to increase. I had to put it on a different scale, an order of magnitude different, just because it is overall higher, but just to show you the relative changes, I've put it on the right axis here. And then there's a dip in 2000, and then if we put in the term soil health in there, I was actually surprised to see this little bump at around the same time that soil tilth occurred. But then you get a lag, and then you get this increase. And even though there was a dip in 2000, I have a theory for that. I can talk to you about it after the talk if you're interested. And then it's continuing to increase. So it just shows, one, the evolution of these terms for something that is kind of more intuitive, but also how one's phasing out in the other and the continued popularity of soil health. Okay, this is something I asked my 354 class. What's causing that bump? And those of you guys that have been in 354, you're not allowed to answer. What's causing this peak here in the late 40s? I don't think so.
Too early.
Yeah.
Post festival.
But I have to say it's fun, but it's a little bit right.
I think John had it right. I don't know if you guys could hear him in the back. He said post-Dust Bowl. I think it's the Dust Bowl. This is when we were really thinking about tilth and health. And of course, there's a lag, right? This happened in the 30s. But there's going to be a lag in when magazines and newspaper articles and even scientific literature stand in this as well. We're using those phrases. So I think that's what drove that trend. But anyways, so moving a little more about soil health. The other way, so I gave you the definitions and function was a part of it. So soil function is just another name for a process in the soil. But there are specific processes that are beneficial to humans that we care about. We call these soil ecosystem services. And these are a pinwheel here from the Food and Agriculture Organization on soil health, or on soil ecosystem services. And many of these we care about as humanity, but we might not care about so much as agronomists. So cultural heritage, right? There's stuff buried in the soil that archaeologists can find and get dating from. And learn about human cultures, past human cultures. Soils are also a foundation for human infrastructure. While that's great, we're in a building that is built on top of soil. But the top ones that I've highlighted here are the ones we care most about as agronomists. So especially provision of food, fuel, and fiber, carbon sequestration, water purification and reduction, contaminant reduction, climate regulation, nutrient cycling I've highlighted here, and habitat for organisms. But especially microbes are important in many, if not all of these soil ecosystem functions, but especially this nutrient cycling one, and for nitrogen, phosphorus, and sulfur, which are mostly found in organic forms. They need to be mineralized by microbes before they can be, those nutrients can be taken up by plants. All right, so I'm going to be preaching to the choir and all this stuff. So bear with me, those of you that might not be familiar with soils. This is an idea soil. 50% of it is solid, 50% of it is a pore space. And that pore space can range between the fraction of air and water, but in an ideal, we might want about 25% of each. The solid part that's on the top here, 45% of it can be mineral particles, and then 5% organic matter. And organic matter is central to soil health. And I'll show some reasons why in a little bit. But out of that 5%, 80% of it is well-decomposed organic matter. That used to be called humus. Now that term is kind of being phased out. And then the other 20% of it can be split up by living roots and organisms. And I'm going to focus mostly on the living organisms in the soil, but that's not to neglect that these roots aren't important. And actually all that decomposed soil organic matter is also important and has an important soil function. All right, so this is what I like to call the living soil organic matter. This is a food web. Down here on the bottom is the residue, kind of the base of the food web. We have microbes that colonize that residue. We have shredders that will shred it into smaller pieces and make it more accessible to microbes. And we have secondary consumers, tertiary consumers. And these are all driving the food web, the soil food web. Those are all components of it. If I were able to gather up all of these things in a soil, 1 acre of soil, let's say I was able to do that, right? That would be impossible to separate out microorganisms from soil particles, but let's say I could do that, and I could put them all in a crate and set it on a big balance. How much do you think an acre of soil, how much would that weigh? Would it, and tell me, raise your hand when you think I've gotten to the appropriate amount of weight here, and then also the animal that's kind of equivalent of that weight. Dogs, so 75, a large dog that's 75 pounds. No takers? How about a human? Average human, maybe 160 pounds in 1 acre of soil, all those living things. Still no takers. How about a bison? So now we're at 1,200 pounds. Still no takers. Bull, 2,000 pounds. You guys all being shy and just don't want to raise your hand? Okay, we've got a taker. All right, Dr. Thompson has raised his hand for the bull. All right, how about elephant? 8,000 pounds. Okay, so the majority of people say at least one elephant. All right, let's look it up. Let's look at these numbers to kind of calculate, come up with that calculation and see if that's correct. Okay, so these are all the soil flora and fauna, or animals and microbes, sorted by smallest to largest. Here's their size in millimeters, their numbers per gram of soil. So that could be anywhere between 100 billion viruses or more to an earthworm doesn't even fit in a gram of soil, right? That's why it's not applicable. And this is the biomass estimates. This is from Brady and Weil in a microbiology textbook that I've used. These are ranges, right? So these are very high organic matter soils to low organic matter soils. We don't have a good estimate on viruses. They're very difficult to quantify. But then we've got ranges, so 400 to 4,500 pounds just of bacteria and archaea, let alone adding up all these other things. And fungi can be up to 13,000 pounds by itself. And what weight? So I gave you guys an estimate of an 8,000 pound elephant. So when we add all these up, the low end is 1,800 pounds, but it could be as much as 27,000 pounds or more. And then if we want to look at that elephant equivalence, most of that is in microbes, right? So we've got almost one whole elephant just of fungi in some soils, probably mostly forest soils. But there's a third of of the soil biology that's in the other microbes as well, bacteria, archaea, and actinomycetes. So if we add all these up, it's actually could be over. So this is an average, or I split the difference here in this range. So it's closer to 2 elephants rather than 1 elephant. But you guys would have guessed that anyways, right? I just didn't have the option of 2 elephants up there. So this is from the Soil Health Institute. They created a video, and I kind of really like this image of going out on an acre of soil, or an acre of farm, and imagining 2 elephants underneath your feet of living organisms, right? Even though it makes up a small fraction of total soil organic matter, these living organisms, in an acre of soil is equivalent to almost 2 elephants underneath your feet. And as I mentioned, that those microorganisms are really one of the major players, at least in abundance and size of biomass. And this is so I adapted this figure from a textbook. So those microbes are actually kind of like the eye of the needle that all organic matter must pass through, right? We know how crucial and important they are, and it's part of the reason why a lot of these soil health tests are either trying to measure microbial biomass itself or activity of those microbes. So all residue must pass through this eye of the needle, so a very small fraction of soil volume. And then that residue in organic matter gets converted to CO2. Some of it can be reallocated to more microbial biomass. Some of it gets stabilized to soil organic matter, which can then be run back through the eye of the needle again, or decomposed by microorganisms. And all of this processing also results in mineralization of that organic matter and making nutrients available to plants, especially NP and S. Okay, so this is a provocative cover, obviously. So this is just a clip I got from Politico, which is an online magazine, but it said, "Can American soil be brought back to life?" What does that imply?
It's dead.
Yeah, it's dead or dying, yeah. I don't agree with that, but it's a provocative title, and it makes for an interesting read. So, but what they're talking about is all these living organisms and microorganisms in soil, and this author, at least to get your attention, is claiming that they're dead or dying. All right, but there are ways. So if we even take that claim at face value, there are ways that we can improve microbial activity or biomass, this kind of central component of soil health. And everyone's got their own lists. Harold Van Ness at Mahdi's conference last week gave a talk on this, and he had his own kind of 4 principles. I think he might have had 5. NRCS has 5 or 6. But these are the 4 principles I've kind of boiled down in order to increase soil health. So reduce disturbance, so minimize or reduce tillage and other disturbances. Add carbon, so just leaving more residue on the ground, adding a cover crop. Manure as a source of fertilizer instead of synthetic manure. That's all going to increase soil health. Perennialization, so make sure there's living roots in the ground between cash crops, like a cover crop, for example, or unprofitable pieces of land, convert it to maybe a perennial crop. And then diversity of plants. This is an area that I've done a bit of research in, looking at diversity of crop rotations, so diversity in time, and effects on soil health. But in short, most of these things all either increase microbial biomass or the activity itself, right? And that's what farmers are looking for, at least, so with soil health tests. So these are reasons why a farmer might use a soil health test. It's a comprehensive assessment of soil's productivity, but also sustainability. I've got some data. I'm not showing it here, but increasing microbial biomass means long-term increases in soil carbon too. So that might be kind of a sustainability aspect. It helps agronomists evaluate effective management practices on soil and provides incentives for keeping soils healthy. Right? So it's a way to get some feedback to see if a management practice you're using is having an effect on soil health. And then in the future, it's not right now, but it may be a way to evaluate land and assess land price, similar to CSR2 or the Corn Suitability Rating 2. It's not right now, but then there's a lot of public-private soil health initiatives that are sweeping the nation. These aren't regulations, so California has a Healthy Soils Plan, Maryland passed a bill, I think there might have been a couple more states since then. And then the Soil Health Institute, which is a foundation, are mostly paid through Noble and Farm Foundation. Dr. Thompson and I were actually at one of their meetings a few weeks ago, and they're doing some interesting stuff, taking a lot of measurements on soil health. So those are all reasons why, when I go out and give extension talks, I talk to some farmers and say, these are reasons why you might want to measure soil health on your farm. These are my lists, well, an adapted list from this paper. In any of these papers, if you email me, I'd be happy to send them to you. This is Doran Zeiss. They wrote a paper about soil health tests, and they said, or soil quality, they were calling it at that time, it needs to be sensitive to variations in management. That's rule number 1. And there's a problem with that because a lot of things we can measure in soils aren't very sensitive to management. And this is a good example of this. So this is some data from my lab, some Reganko sitting in the back. This is data he's been working on in collaboration with Larry Siacek, who's at North Dakota State University, but he collected over 250 soil samples at different landscape positions in differing years of being in CRP. So 0 up to 40 years being in CRP. 0 is just business as usual, corn, soybean rotation. So we'd expect after 40 years, or at least I thought, or we expected, that we'd see an increase. But there's a lot of variability, right? So these are, each one of these is an N of probably 10 to 20. And this is soil organic carbon in grams per kilogram of soil. And this out here, obviously, I have it labeled here as native grassland. And this is the only soil that was significantly greater in soil organic carbon than the corn-soy rotation. And I forgot to mention, these are soils from northern Iowa, western Iowa, southern Minnesota. So a lot of them in the Des Moines lobe. We've tried to keep the soil types the same, and again, variation among landscape position. And we couldn't detect a change even going out to 40 years. Although you see a bit of an increase at least at 40, but you definitely don't at 25. However, If we look at something that's more of a measure of microbial activity, this is potentially mineralizable carbon, so this is similar to the Haney test, where you wet up a dry soil, you measure the amount of CO2 that's respired. We happen to do a 100-day incubation, right, Mercado? This is about a 100-day incubation. So we did do a bit of a bit longer incubation, but these are how many milligrams of carbon per kilogram of dry soil was respired by those organisms. So it's an indicator of microbial activity, their biomass, and in this case, we actually see at about 19 years, there's a significant difference than the zero, the business as usual. So this would be a good soil health indicator, at least for soils that we were working in. This, not so much. We couldn't detect management differences early enough. All right, so it needs to be sensitive to changes in management practice. So I showed some evidence of why that might be the case. It needs to be well correlated with beneficial soil functions or those soil ecosystem services I was talking about. All right, so I'm going to go through kind of a little hypothetical example here, but I'm going to show some data from Alan Franzluber about this later. So this is how many or how much fertilizer nitrogen needs to be added and the percent of maximum yield. So there's two soils. Soil A here, where we get this kind of exponential increase to a maximum, and soil B, where we don't see any response. It's at 100% without any nitrogen rate. Which soil would you rather have? B, right? Yeah. You don't have to add any nitrogen to it. And this has been one of the most compelling links of soil health to a function like, and what this function is the nitrogen supplying power of soil B, right? And you're saving money on fertilizer. And I'll show some data. John Sawyer and others have done this in Midwestern soils too, but I'll show some data in a little bit that goes into that in more depth. So these other lists of ingredients are cause and effect must be known, so both the cause of a low test value and then how it relates to that beneficial function, like maybe nitrogen supplying power. It needs to be comprehensible and useful to land managers, so what can you do about it? And then this is an important one. It needs to be easy and inexpensive if you're going to make it a test, right? Because these labs, they get high volumes, they need to do it very quickly, and they have tight margins, so they need to do it inexpensively too. And this is what I want to focus on. Has been part of the citizen science aspect of the research we're doing in my lab too, is finding easy and inexpensive soil health indicators. So this is a diagram I borrowed from Dr. Alcasi in the back there, and David Kwamenza. This is in a extension literature they had. It's basically a triangle of soil health, and the indicators that fall under physical category, chemical, biological, and soil organic matter is really the central to all of them, because if you're raising soil organic matter, you're also decreasing bulk density, you're increasing porosity, improving soil structure, aggregate stability, but you're also increasing CDC, total carbon and nitrogen, under these chemical indicators. pH is typically better. And then the biological, because soil organic matter, or the biology is actually part of soil organic matter, and also fed by it, potentially mineralizable carbon and nitrogen are good indicators. Measuring soil microbial biomass itself, microbial activity. And then some of these, we don't really have a good idea about what a good number should be. So things like microbial diversity, we don't know how to relate these to a known soil function, like nitrogen supplying power. But people are still measuring it, and some soil labs will measure it and give the information to a farmer. And then decomposition is one I've added here as well. But these all should really relate to improved yields, as Madi's extension document pointed out as well. If you're using these indicators as some measure of improved soil health, they should also correlate with improvements in yield. All right, so I hate to pick on these labs, but I often do this when I talk to farmers or students in my classes as well. So I picked out 4 labs. These are common labs that have soil health packages. And they usually have more than one package. They have a basic and then an extended. I always find it ironic that Cornell's comprehensive assessment of soil health is called CASH, and it's also the most expensive. So these are the costs over here. So from 45 to 65 for Ag Source, Midwest Labs, 55 to 65. But you could pay as much as $170 for the extended Cornell soil health test. So, I mean, that's a lot of money, right? I mean, you don't have to be a farmer and know that that's not very feasible. So if we average these labs I've kind of picked on here, it's about $78 per test, but the range is $45 at the minimum to $170 a sample. So again, not very conducive to encouraging farmers to test their soil for soil health. And you guys can see where I'm going. Did anybody see the CSA news article last February? Raise your hand if you saw this. It was about citizen science and agriculture in general. So there's a lot of, most citizen science, I would say, with regard to agronomists and soil scientists, takes place with strip trials. And I work with some farmers, some practical farmers of Iowa that are trying out strip trials. They might be taking their own yield measurements or cover crop biomass. So that's probably the most common type of citizen science.
Water quality testing.
That's true. Yeah, water quality testing is now, Laura's been doing a lot of that. Like the way you participate can actually take place at many different levels. So it could be collecting the data, it could be sharing records, providing access. So when Teresa goes out and does a lot of her measurements, they're just providing access in many cases. But we also are working with farmers that are collecting some of their own data and informing experimental treatments. And so I'll talk more about the kind of benefits and costs of working in citizen science. But this was a good article. If you have a chance, go back into archives. You could get these if you're a member of the Tri-Societies. I think you can even get these and not be a member, right? They're available to the public. If your university carries the license, I think. So, this is again Marshall McDaniel's list of soil health DIY measurements. Some of them you guys might know just from interacting with my grad student or me. So there's an aggregate stability test. I'll show a little bit about that. A bulk density test. And I have the citations. Again, I'm happy to send these to you if you're interested in any of these. Decomposition. I'll talk a little bit more about that one, of course. Earthworm midden count is a way people can measure earthworm activity. Soil erosion potential. So Matt Liebman and Tim Youngquist are actually doing that at the prairie strips. Some neat citizen science work there. And then Solvita sells these respiration paddles that are used in the Haney test. And then water holding capacity, there's some easy ways to do that. But these are all indicators under that triangle I showed. But these are methods that farmers can do themselves instead of paying $45 up to $170 per sample, right? And so these are some of the functions being measured. So ease of root penetration, earthworm activity, biological activity, nitrogen supplying power. The erosion pads measure soil loss from the field. This is sheet erosion. But you can see a common trend in all these, the equipment you need. You really only need a two-place balance. And nowadays you can get a pretty accurate one that goes out 2 decimal places cheaply on Amazon. I do have to caution you though, I had one farmer at a talk I gave say he got in trouble, he ordered a couple balances because I guess they thought he might be cooking meth or something in his. So they put you on an FBI watch list if you order too many of these. So be careful about that. That was a pretty good laugh when he told me that. But yeah, you know, there's some other kind of cheaper materials involved in some of these, but really all you need is a two-place balance. That's the common denominator for all of these measurements. Okay, soil erosion pads. So I mentioned this. This is work that Tim Youngquist and Matt Liebman are doing at the prairie strips, and maybe they're doing this in some other experiments too. But these are just nylon screen door mesh with another type of nylon stapled together. And they use toothpicks to pin it in the soil. And then they leave it out in the field for, I think they leave it out for a couple months. And the data I'm showing down here, these are 5 different prairie strip sites. And they have the control, and this is how much soil movement in tons per acre per day. And then the treatment, or the paired farm or watershed that had these contour prairie strips to prevent sheet erosion. And what Tim and Matt found was that they did see a significant decrease in soil movement thanks to those prairie strips. And they've done measurements that weren't citizen science related that have also confirmed this. They used some H flumes at the bottom of these watersheds and prairie strips to show that they do decrease erosion, soil erosion. All right, so aggregate stability. This is a measure that Jeff Herrick came up with. Kind of a quick and easy way to do this. It's published in the journal Catena. So you go out and you dig a little trench with this tool that they provide you in this kit. I'm sorry these pictures didn't come out that great. And then after digging down about an inch or two, you excavate an intact soil aggregate that's just below the surface, and you put it in this homemade PVC cup with a mesh on it. Again, these are all very relatively inexpensive materials. And then you could get these fishing tackle boxes that have water in them, and you dip the aggregate in there in a sequence, and you can determine how stable these aggregates are in this soil by just dipping it in this kit. And then there's a data collection sheet as well. So here's an example. So this was a soil that had very low stability. This is what it looked like before they dipped it, did this series of dips in water. This is after the dips. This is a highly stable soil. This is original. I know it's hard to see that aggregate there, but then after 5 dips, it stayed intact. So that's another DIY soil health measurement. The one I'm going to focus the rest of my talk on is decomposition and respiration. So respiration, and they're both, I always tell people they're both 2 sides of the same coin, right? So earlier I showed the soil food web. One way we can determine the activity or the abundance of all these organisms in the food web is to measure how much CO2 they produce, right? Many of these organisms are like us. They're heterotrophic. So they breathe in oxygen and they exhale CO2. And they need sugars as well, or a carbon source. And the most common do-it-yourself way to measure this is with the Solvita. And it's commonly used in the Haney test as well. And I don't have to go over that equation for you guys. I've seen that in school. But I do want to mention that even though this is do-it-yourself, it's really not that inexpensive. And those of you that might have purchased these, I use this for educational purposes, but it's over $13 for each one of these paddles. If you want to get a more precise reading of these paddles, you have to pay this paddle reader. Does that price look right, Dr. Thompson? I know you have a couple of these. So over $1,500 for sure. That was the last time I looked into that price. And so what does that information give you? What does that CO2 give you? Well, it goes into this Haney score, this Haney test score, which is derived from these 2 Haney papers, which they go over in extraction. So some of the extraction is part of this equation as well. And this Franz Luber paper that they show CO2 released in a 24-hour incubation. He's actually moved now to a 3-day incubation. Again, I'm happy to send you these papers if you're interested in them. But you end up with a score that ranges from 0 to greater than 25. The more CO2 respired, since this is a large driver. I won't go into where they get these numbers in the denominator because it's not even clear from these 3 papers. But I do like this paper. So last year, Alan Franzuber had 3 trifecta papers. And this was from the 3rd of the 3 papers. And he did nitrogen response trials. I think it was over 80 soils around North Carolina. So this is kind of like that hypothetical graph I showed before. So we have how much nitrogen needs to be added to bring to optimal yield and two soils. So again, you guys said you'd want soil B. Here's soil A. This is the corn grain yield. This was at 100%. This one required nitrogen to get to max yield, right? So what Alan did, and I find this pretty compelling, is that he took that biological test activity at 0 to 10 centimeter depth, and he correlated that to the economically optimum nitrogen fertilizer rate. And so that's just pounds per acre. So this soil, the EONR would be about 200 to reach maximum. This soil would have an economically optimum nitrogen rate of 0 because it's already at maximum. And what Alan showed is that the greater the soil test biological activity was, the less nitrogen you had to add, right? So it seems to be a pretty good indicator. There's some noise. There's a lot of zeros and spread of these zeros down here, and he's picked out some of these outliers. These squares were outliers he'd taken out. But that seems like a pretty good evidence. And then Sawyer and others published a paper last year that I don't think they had as many sites, but they used this biological activity and correlated it with the ONR as well, found an okay correlation. Okay, so that's great. Respiration's great. At least gets to nitrogen supplying power of the soil. But as I showed, it's expensive, right? And that respiration measurement can sometimes be variable as well. There's some other issues with it. So one thing my lab has been working on is, like I said, the respiration and decompositions are both sides of the same coin. What Teresa's been working on is working at measuring decomposition of the mass loss of materials as it moves through the food web. And this isn't a new idea. I mean, I didn't come up with this out of the thin air. It's actually been around for a long time, and this is one of the earliest references I could find to decomposition being a good measure of fertility or health of the soil. So this is in J.I. Rodale, so you had brought up the Rodale Institute, part of the organic farming movement, and this is in a farmer that's quoted in his book, Pay Dirt. And this farmer said the soil is so healthy, they said fertile, but I inserted healthy there, that it would eat almost anything thrown at it from a gunny bag to corn stover. So this is kind of an intuitive thing farmers have been seeing. You know, a healthier soil, you get faster decomposition of residue. And this, of course, has led to the Soil Your Undies trend on Twitter. So this is a demo that Neil Sass did up at the Soil Survey Office in Waverly. He had buried these underwear for 2 inches in the ground for 6 weeks in conventional corn, no-till. And this is the percent that was decomposed. No-till soybeans with cereal cover crop, cereal rye cover crop, permanent pasture, and then the alfalfa is almost disintegrated. It's nothing left except the elastic band. So we've been doing that. I do that for demonstrations at farms. So here's one I did at the Southeast Research Farm. I talked to about 50 farmers that came out to visit for a field day there. I had the staff out there bury a couple pairs in 30-year no-till, 2 years of cover crop, and then the business as usual, conventional chisel plow in the fall, and no cover crop. And you could see, just visually see the differences, and it makes for a good laugh too, right? I mean, this is pretty funny and compelling. Makes for a good demonstration, but it's not a very good, robust indicator of soil health, right? That's, as a scientist, I'm very critical of it from that angle. And it's messy and very inconvenient to retrieve the underwear. So when we've tried this, the cotton will kind of peel off on aggregates, so it's hard to recover all the cotton. Soil temperature and moisture are often stronger regulators of decomposition than something like microbial biomass or that nitrogen supplying power soil. And this adds a lot of variability, but this I think is also a problem with the Haney and Solvita as well. Then it makes it difficult to compare among soils and treatments if the temperature and moisture is really the driver of the difference rather than microbial biomass or the nitrogen supplying power, right? So the question is, sorry this red sometimes doesn't show up very well on the slides. So can we develop a scientifically robust yet inexpensive soil health indicator involving decomposition. So the decomposition thing is key because, one, it's measuring that other side of the coin, the biological activity, but it's also inexpensive compared to measuring CO2. So this is where Teresa's work comes in. So she had 2 years of collecting data, and she'll talk about on March 6th. In 2017, we buried tea out in 9 farmer-run strip trials of cover crop versus no cover, and then 1 long-term ISU cover crop farm that's been in 16 years of cover crop, no cover crop. Last year, she did 8 long-term ISU farms comparing business as usual, so soil health degrading practices versus soil health promoting practices. Some pretty neat experiments in there. Some of them you might be familiar with. The Cobb site that Dr. Thompson is a lead PI on. The Marsden site that Matt Liebman is a lead PI on. And so, again, these are a little bleached out. I apologize for that on the screen. But we're looking at these 4 kind of household substrates as biological indicators right now. So green and rooibos tea, the advantage of these is that they're in self-contained litter bags already, these nylon bags. We're using the underwear again, just kind of testing it out, even though it seems to not be a very good indicator. And then these birchwood craft sticks, it shows the carbon to nitrogen ratio of all these materials, the target organisms. So these are mostly microbes because soil fauna, mesofauna can't fit inside the mesh holes in the tea. Whereas the cotton incorporates all organisms, including those shredders. And then the birch craftwood sticks, mostly fungi, because there's a large concentration of lignin in those sticks. That needs to be broken down by mostly fungi. This is how many days in the field we left them. And then to give you a comparison to the Solvita test and some of those Haney tests, how inexpensive they are compared to those tests. So even if these aren't perfect, they're still a lot more affordable for farmers to measure their own soil health tests, right? And to get the lowdown on that, I'll have to, again, defer you to see Teresa's talk in a couple weeks, where she'll be showing some of the data we found from those 2 years of data collection. So where this is going in the future, I think what we need is an app that can aggregate all this stuff. And I've been talking to Dr. Miller about this. And there's one that Jeff Herrick is already using, and they're starting to get interested in soil health. You can see up here they have coming soon land management and soil health modules. But this is called Land PKS, and it has a lot of functions, but you can, it's based on web soil survey data, but you can also get more objective coloring than using the Munsell color chart. I think they have a texture method on there, so you can do your own texture and enter data into this large database on in their app. But that's where the future of citizen science really needs to move, is there needs to be some kind of accessible application or something like that. Okay, so the benefits of participatory science or citizen science: you can engage and you can inform stakeholders. In my case, a lot of times we're talking about farmers. This is a good way to engage farmers in measuring their own soil health, but thinking about it. Power numbers, so we can gain knowledge of larger phenomena by including more people and measuring it in more locations, right? We as faculty and students and postdocs can only measure so much at one time. Some of the limitations or things that make it challenging is coordination, consistency of data collection, and it can be tricky. The quality assurance, quality control of just having anybody measure data, of course, is more of a concern than coming from a trained graduate student, a well-trained graduate student. And then trust between the data collectors and the data aggregators is really needed, right? You're measuring data, you're giving your data to someone who aggregates it, synthesizes it, should that data be given back? And how is it used? So that's going to be one of the limitations and challenges. And then that's it. That's really all I wanted to mention about soil science and participatory soil science, citizen science. And these are all the folks I'd like to thank, including many I couldn't find on the list, some of the long-term experiments. We collaborated with PFI on a lot of the measurements, especially in 2017. And the funding for Teresa's work came from the Leopold Center of Sustainable Ag. And at this time, I'm happy to take any questions. Danny?
Well, at the starting, you were kind of going through the terms soil till, soil quality, and soil health. When you said you might mention it later, why there was that late dip in soil health.
Yeah.
I'm just curious why you did that.
Okay, good, you were paying attention. So Danny was talking about that dip in 2000, and if you notice, all three of those terms dipped quite a bit, like a relatively large amount. What happened in 2000, 2001 that caught the world's attention? You shook your head, you know what it is.
Yeah.
What is it?
It went down.
Yeah, 9/11. I think anything not related to terrorism or 9/11 probably went down a little bit, dipped at 2000. That's my theory. I don't know for sure. Again, it's Google won't let me access all their data, but I think that's why. Way to pay attention to that. Yes, Laura?
Could you talk about your new project involving citizen science, but specifically with American Indian communities?
Yeah, thanks for bringing that up. So I'm working with an anthropologist, and she's been working with Native American communities for a good amount of time now. That she specializes in Native American communities. And one of the concerns they have is measuring soil health in these community gardens. And they practice this traditional practice of intercropping called Three Sisters. So planting common beans, squash, and corn together. And so Christina and I are trying to engage Native American farmers and gardeners in measuring their own soil health in these gardens. But more specifically, we're going to be using some of these decomposition measurements, some of these do-it-yourself soil health tests, and we're going to get them to do some kind of replicated trials of the Three Sisters by themselves and then together to see if we can see effects on soil health within a short amount of time. I'm excited about that project, by the way.
And it's in the Midwest, right?
It's all with Midwestern Native American communities, yeah. So the Omaha, Oneida, Meskwaki is the closest one. We just hired our first graduate student, so he'll be working with, and he's an agronomy undergrad, and he's going to be housed in anthropology and dual sus-ag major. Derek Kapayu, if any of you know him, he's a good student. He was in my 354 class, so he'll be collecting those data. You'll have to ask him for the updates on that project once it gets started. Dr. Manu.
Yeah, I saw in your decomposition trial that Teresa is doing. I realized you had the different days, number of days that you let those things come. What is the principle behind that?
Yeah, great question. So with the T, we see that the mass loss stabilizes at that point, and we're actually trying to stick with the method that some folks in Europe were using. So that's the rationale for that. So, you know, there's this kind of exponential decline and then the mass loss stabilizes. So this is when the mass loss stabilizes, usually about 60 days or so. The cotton, we base that upon just we know how quickly it decomposes. You don't want to leave it out too long, but you need to leave it out long enough to decompose and all the treatments you're doing. And most people do between 4 weeks to 6 weeks for that. And then we knew this would break down the slowest, just based on the C to N and the amount of lignin. So we left that out, the very last T. So the T was the only one that we pulled out at multiple time points to get an exponential decay. But this one we just pulled out at the end. We excavated it at the end of when all the T was done. But Teresa will talk more about that on March 6th.
Thank you.
Any other questions before I have to run the class? Otherwise, you guys know where I'm at. You can always drop by my office. I'm happy to chat about this stuff.
I just have one small note that this cost per sample is only the cost of the material, but it does not include the other charges if it is offered as a test method, routine lab.
Yeah, you're right, that doesn't include labor.
So it doesn't include labor or in some cases infrastructure and
But-
other things.
But neither does the CO2. That doesn't include labor. You have to do this
Yeah.
yourself.
Yeah. This is the cost of the equipment.
Yeah. Yes, Fritzy.
I'm just curious, for the participatory soil health tests, and you mentioned that you have promoted this to the farmers.
Mm-hmm.
I wonder how receptive were the farmers about this soil health test, because now we have to think about soil fertility tests, and now we have to add soil health tests.
They're pretty receptive about it, but again, most of the farmers I talk to, there's a lot of excitement about it, especially our decomposition work. The farmers that I first presented the idea to, they wanted to order tea right away. They were so excited about it, and I mean, they were chomping at the bit. Because they realize the power of measuring microbial activity, right? And they want an inexpensive, easy way to do it on their farm. So I think it's really powerful and can capture quite a bit of attention. Some of these other tests, they're a little more labor intensive. So bulk density is not too bad, but that aggregate stability test isn't too bad. It'd be neat to do a column of how much labor hours these would all take too. The erosion potential would take some time to put those together for those mesh pads. The earthworm emitting count's pretty easy. You just make a PVC quadrat. Yeah, some of those are. I think there's even though farmers are busy, I think they are interested in measuring their own soil health. At least the ones I interact with.
Well, just to follow up on the question. Now, if a farmer is doing it just for curiosity, he's not going to get the result that he's going to apply to his farm to increase its yield or something, or just for the curiosity.
I think it's curiosity is driving a lot of it. But if one of these tests, well, the respiration, I've already shown you the data from Franz louvers. That's a good indicator of nitrogen supplying power. Now, we're not at the stage of making any recommendations about it yet, so labs won't make recommendations about it, but at least the data that both he and Matt Yost and Sawyer and others showed are really compelling. Where did I have that at? Oh, sorry. Went the wrong way. I mean, that's the next step, right? I think curiosity is driving a lot of it, but at some point, we might see recommendations, nutrient recommendations from a test like this. Yes, sir.
If the data are going to be useful for farmers, they're going to want to track these data over time. And one of the things that we mentioned at the very beginning was that the first criterion for judging value of a test is its sensitivity and response to management.
Mm-hmm.
One of the things that determines sensitivity that we all know is the qualifications of the person doing the test and how much time they have and whether they're getting paid for it or not, and how they collect the sample. I just wonder if the sensitivity of these tests that are performed by citizens as opposed to, as you said, trained professionals, is going to be at the same level that will allow them to track over time the changes that are going to be.
I think the quick answer is no, immediate, that it's not going to be as good as a well-trained graduate student collecting these data, right? But over time, especially with training, citizen science data could be just as robust as professional scientists or traditional scientists. Laura, what do you think? You've been
Well,
collecting a lot of
and
these data.
I can't cite any examples off the top of my head, but I've tracked literature with water quality testing that is comparing scientists with citizen scientists and advocating for, say, certain types of tests for certain nutrients that, quality can be high. But I think you're totally spot on to talk about quality assurance through something like appropriate training. And then, of course, there are the things of, lack of follow-up or follow through over a long period of time that we know is part of doing good science.
Yeah, and there's all kinds of sources of variability too, right? Not just human, but climate variability is going to be thrown in there too. It's all about finding the signal in the noise, and the more measurements you make, the more likely you are. Yep, long-term. Yeah, I would advocate for long-term experiments.
Trying
for
to
sure.
control sources of variation that are would be noise. Confounding interpretation of results.
Great. Well, I'm sorry, I got to go. I'm actually running late for my next class.